In this video, we'll focus on the last stage of cellular respiration, known as oxidative phosphorylation. In the fourth stage in cellular respiration, the high potential energy of electrons carried by NADH and FADH2 is gradually released as they move through a series of redox reactions. The proteins involved in these reactions are called electron carriers and form part of the electron transport chain. These proteins are located within the inner membrane of the mitochondrion. After the electrons pass through the electron transport chain, they are accepted by oxygen, which combines with protons to form water. If you look at the energy diagram for the electron transport chain, where we're measuring the free energy change relative to oxygen, we can see that NADH and FADH2 donate the high energy electrons that they're carrying to different complexes within the inner mitochondrial membrane. These electrons flow through the electron transport chain and are finally passed on to oxygen, which combines with protons to form water. The energy that is released as the electrons flow through the electron transport chain is used to create a concentration gradient of protons across the inner mitochondrial membrane, which is then used to drive ATP production by ATP synthase. In concert with electron movement through the electron transport chain, protons are pumped across the inner mitochondrial membrane into the space between the inner and outer mitochondrial membrane, known as the intermembrane space. This forms a strong electrochemical gradient across the inner mitochondrial membrane. These protons then pass through the enzyme complex called ATP synthase and drive the production of ATP from ADP and inorganic phosphate. This process links the phosphorylation of ADP with NADH and FADH2 oxidation, which is why this stage in cellular respiration is known as oxidative phosphorylation. Most of the molecules that form part of the electron transport chain are proteins containing chemical groups that facilitate redox reactions. All of these proteins except one are embedded in the inner mitochondrial membrane. One of them, called ubiquinone, can move throughout the membrane because it is lipid soluble. Both NADH and FADH2 donate their electrons to different complexes within the inner mitochondrial membrane. NADH donates electrons to a flavin-containing protein, whereas FADH2 donates electrons to an iron sulfur protein that passes electrons directly to ubiquinone. As the electrons pass through the electron transport chain, protons are pumped across the inner mitochondrial membrane from the mitochondrial matrix into the intermembrane space. ETC proteins are organized into 4 large multi-protein complexes and cofactors called complex 1 through 4. The proton pumping action is contained within complexes 1 and 4. Ubiquinone and the protein cytochrome C transfer electrons between complexes. Ubiquinone also carries protons across the membrane. If you look at an image of the 4 complexes, it is important to note that NADH donates its electrons to complex 1, whereas FADH2 donates its electrons to complex 2. Both of these complexes pass on their electrons to ubiquinone, which then passes the electrons on to complex 3. Complex 3 then passes the electrons via cytochrome C to complex 4. As the electrons are passing through the electron transport chain, complexes 1, ubiquinone, and complex 4 pump protons or hydrogen ions across the inner mitochondrial membrane into the intermembrane space. This creates an electrochemical gradient of protons with a higher concentration of protons in the intermembrane space compared to the mitochondrial matrix. The final electron acceptor is oxygen, which combines with hydrogen ions or protons to form water. The energy stored within the electrochemical gradient of protons helps power ATP synthesis via the ATP synthase complex. A protein complex that we haven't discussed so far is the ATP synthase, which is the final component important for the production of ATP. ATP synthase is an enzyme complex consisting of two components, an F1 unit and an F0 unit. The F1 unit, known as the knob, has ATPase activity, whereas the F0 unit is a membrane-bound proton-transporting base, which enables protons to move from the intermembrane space back into the mitochondrial matrix. These units are connected by a rotor that spins the F1 unit, that spins the knob, and a stator, which interacts with the spinning F1 unit. So this ATP synthase complex is extremely large and contains many subunits that work in concert with each other to bring about the synthesis of ATP using the energy stored within the electrochemical gradient of protons across the inner mitochondrial membrane. So in a sense, the ATP synthase is like a motor, but it's a molecular motor where protons flow through the F0 unit, leading to the spinning of the rotor, which in turn spins the F1 unit. And when the F1 unit spins, the subunits in the F1 unit change shape, catalyzing the phosphorylation of ADP to ATP. The force that drives the protons through the ATP synthase complex back into the mitochondrial matrix, thus powering ATP production in this way, is called the proton motive force, which serves the basis for the chemiosmotic hypothesis. Thus, oxidative phosphorylation is the stage where the vast majority of the payoff from glucose oxidation occurs. ATP synthase produces 25 of the 29 ATP molecules produced during cellular respiration. Thus, the oxidation of one glucose molecule leads to the production of 29 ATP molecules. So if we start with one molecule of glucose, by the time we get to the end of cellular respiration, we have 29 molecules of ATP, most of which are generated by the ATP synthase complex. Oxygen is very effective as an electron acceptor because of its highly electronegative nature, meaning that it holds on to electrons tightly. Since there is a large difference between the potential energy of the electrons contained within NADH and oxygen, this allows for the generation of a large proton motive force needed for ATP production. There are other organisms that live in oxygen-poor environments that use different sources of carbon other than glucose, or different electron acceptors like nitrates and sulfates. As a result, since they're not as efficient as oxygen, those pathways lead to a lower yield of ATP at the end of cellular respiration in those organisms. Cells that don't use oxygen as an electron acceptor and grow in anaerobic conditions thus grow much more slowly than cells that can use oxygen. This brings us to the end of our discussion about oxidative phosphorylation. After going through this information, you should be able to respond to the questions listed on this slide. Thank you.